Hello, everyone. Steph Fradette. I work at Biogen. I oversee our neuromuscular portfolio, but what's most near and dear to my heart is I was the clinical development lead for the tofersen program. It's an honor to be here today. I have a very special place in my heart for all of the work that ALS TDI does. And you'll see a couple of examples where a data set like the ARC data set could have been incredibly valuable to make better decisions on the tofersen development program many years ago. So I'm really looking forward to what that will do for drug development going forward. A couple of important disclosures. Like I mentioned, I am an employee of Biogen. So I was thinking about what to talk about. I always worry that people will get sick of hearing the tofersen story. So I decided to take a little bit of a different approach and try to bring you on a journey and have you sort of walk through what this experience was like for us, but of course happy to answer any questions. This journey started 30 years ago when Drs. Brown and Tipu Siddiq identified the relevance of the SOD1 gene in ALS, first gene implicated in ALS. And about a decade later, Richard Smith and Dodd Cleveland, of course Frank Bennett and Tim Miller, began work on developing the first ASO to target SOD1. And 30 years after the relevance of that gene was identified, we find ourselves with the first genetically targeted therapy approved for ALS. And this journey has been one that probably many of you in the room have been involved in at some stage, and it wasn't all rainbows and butterflies. It's still not, but it's taught us so much, not only about the underlying pathophysiology of SOD1 ALS, but so much about how to run proper clinical trials and really run the right experiment to get an answer to the scientific question that you're trying to answer. And so I'm going to spend some time on the latter half of this timeline, but again, it was a 30-year journey in the making. Before I jump into that, I think there's some important context because you'll hear me talk about neurofilament on almost every slide for the rest of this presentation. So I thought it would be helpful to just take a moment to sort of set some expectations there. But neurofilaments are intermediate filament proteins. They're part of the cytoskeleton of neurons. And when the axons of neurons are damaged, when are injured, they are released into the interstitial fluid and they pass into the CSF and blood, and we're able to measure them. They're a non-specific marker of neuroaxonal injury. I think when we first started talking about neurofilament and ALS as a team at Biogen, I didn't even appreciate how much had been done by the ALS community to really characterize the behavior of neurofilament in ALS. This is probably an underrepresentation of the amount of literature that's out there, so if you know of additional publications, I'm always interested in keeping this current. But there's been so much work, and what we know is that neurofilament levels are among the highest in ALS, people living with ALS relative to other neurodegenerative diseases. And what these researchers have shown time and time again is that neurofilament levels are prognostic for disease progression and survival. So the higher the level of neurofilament, the faster progressing the disease is, the shorter survival is expected. On the left-hand side, you'll see an example of that on a Kaplan-Meier curve. So those with a neurofilament level that was above the median in this study, in green, experienced prolonged survival relative to those with higher levels of neurofilament in yellow. One of my favorite studies I talk a lot about is a study that was conducted by Alexander Thompson and Martin Turner and colleagues. It was published last year. And the group looked at a number of different characteristics that we often use to stratify study populations, enrich study populations. And they found that plasma neurofilament levels, and specifically neurofilament light levels, were the only variable that they assessed that independently predicted survival. So this alone led us to do a lot of soul searching about how we designed our study, which I'll come to in a moment. And a bit of background on SOD1 ALS, which I think is quite familiar to this audience, so I won't spend too much time here, but SOD1 ALS affects approximately 2% of the ALS population. And although it's exceptionally rare, it is also exceptionally heterogeneous. We have over 200 pathogenic variants that have been identified to date in the SOD1 gene, and disease durations across these variants can range from less than a year to over 20 years. Even disease durations within a given variant can be quite variable, which makes drug development quite challenging and fun. The median survival in SOD1 ALS is estimated to be about 2.3 years. For people with SOD1 ALS, mutations in that SOD1 gene lead to production of a pathological form of SOD1 protein that's ultimately toxic for the motor neurons and leads to weakness, loss of function, and ultimately death. But we understand what's causing the disease, and that in itself is a bit of a rarity. I would say we have a relative understanding of what's causing SOD1 ALS, and so we can hopefully do something about it. We can develop therapies that are targeted to address that underlying pathophysiology. And so tofersen was designed to do just that. It's an antisense oligonucleotide, and it was designed to bind and degrade SOD1 mRNA, so the instructions for making SOD1 protein, to reduce the synthesis and the accumulation of that protein and allow the body's natural clearing mechanisms to clear the existing SOD protein, hopefully then preserving the motor neuron integrity. We also have the luxury in SOD1 ALS of a reliable mouse model to assess efficacy preclinically. So in G93A transgenic mice, we were able to administer tofersen and found that neurofilament levels were reduced, so again suggesting a slowing of neuroaxonal injury. Compound muscle action potential was preserved, weight was maintained, motor performance was maintained, and survival was prolonged. So with those preclinical data in hand, and again, confidence about what tofersen was intending to do, we started a single and multiple ascending dose study, where we assessed doses ranging from 10 to 100 milligrams. And as you'd expect, the top dose tested, 100 mg, achieved the highest concentrations of tofersen, and it also achieved the greatest reductions in the amount of SOD1 protein in the CSF, so providing an indirect way of looking at target engagement, and the amount of neurofilament in plasma, suggesting a slowing of again, neuroaxonal injury. We also saw clinical signals, exploratory clinical signals in this study, suggesting that tofersen was slowing clinical decline. And I would argue we may have overinterpreted those data as a company and as a community. I think that we got excited, and ultimately the numbers were small, and the duration of treatment was short. And those data, and particularly the behavior of the participants that were receiving placebo, informed the design of the Valor study. And I'll speak a little bit more to that on subsequent slides. So Valor was a phase 3 randomized, double-blind, placebo-controlled study. It was started in March of 2019, and we enrolled 108 participants around the globe. It took us about 2 years to enroll 108 participants. And again, leveraging sort of what we had learned from that phase 1 study, the availability of natural history data in SOD1 ALS was admittedly quite limited. We thought that we could identify a subset of participants that were likely to progress quickly in the absence of an effective intervention. So you would be able to assess decline in the untreated arm over a relatively short study period, so a 6-month study period. And so we went about doing that and enriching our primary analysis population based on the specific SOD1 variant type and based on the pre-randomization ALS-FRSR decline. So the rate of decline on the ALS-FRSR from symptom onset to the time that they were screened and randomized in the study. We'll come back to these endpoints. The primary endpoint was, not surprisingly, the ALS-FRSR, and it was assessed via the joint rank test in that primary analysis population. And you'll see a number of secondary endpoints listed here, perhaps a little unconventional to have biomarkers as your top key secondary endpoints. But again, leveraging what we had understood from earlier studies, those were the endpoints that we were most confident in and that we understood the biological relevance of. Again, recognizing that the study duration was short, we also had an open label extension study that has proven to be absolutely invaluable to the tofersen program. Participants that completed Valor had the opportunity to enroll in this extension study, which is ongoing as of now, expected to complete in the middle of next year. And we also prospectively planned to integrate data from these studies to understand the longer-term effects of tofersen, comparing early initiation, early start tofersen, so those that initiated in Valor, to delayed start tofersen, those that initiated or had the opportunity to initiate 6 months later in the extension study. The primary endpoint. So in August of 2021, some of you have heard me tell this story, my statisticians called me and said, "Steph, we have the primary endpoint." And they said the p-value is 0.97, and I laughed and I said, "There's no way." Very funny, very funny. And they said, "We're serious." And I'll never forget that moment because I couldn't reconcile what we understood about the biology and the mechanism and what we had seen on the biological sort of measures in the phase 1 study, and that p-value. It doesn't get worse than 0.97. And so it was a long day, a lot of soul searching, but soul searching without other data, which made it even more challenging. And so over the next couple of days, we asked for more data. we dug our sort of, sunk our teeth into everything we could get our hands on. And first we looked at the biomarkers. Did we see the same thing that we saw in earlier studies? And we did. We saw that SOD1 levels in the CSF were reduced by 35%, as we'd expect with tofersen, and neurofilament levels were reduced by 50 to 60% with tofersen administration. We looked at other secondary outcome measures like cell vital capacity and saw that everything was trending in favor of tofersen, but again, not statistically significant by any means. And perhaps for me personally, most importantly, were these exit interviews that I've recently started mentioning, and I think we need to publish because they're so important. This was a qualitative exit interview project that was recommended by the FDA, and we inserted this sub-study into the program very late in the game, so we only had about a dozen or so of these interviews. But we were able to ask participants through qualified experts that conduct these interviews, how has ALS impacted your life, but also how has that changed through your participation in the study? And about 2 or 3 days after the Valor primary was shared with me, I sat down and I read all of these transcripts. And that was the moment that I realized that it was perhaps not the drug that failed, it was the study design that had failed. Because reading the words directly from these participants and their caregivers who so eloquently expressed how the disease had impacted their lives, and not just how that had changed during the course of the study, but exactly when. My disease didn't change for the first 2 to 3 months, and then my disease progression changed. And then I looked at the biomarker data, where it took 3 to 4 months for us to reach maximum biological activity, and everything started to sort of become more clear. So given that prior plausibility and all of the data that we had in hand, we did a lot of work to try to understand what happened with that primary analysis in particular. And I could talk about this for many hours, so I'm going to keep this rather succinct, but I would argue that there are 2 primary learnings that affected the primary analysis in the case of tofersen. First is the approach that we took to control for heterogeneity, so that enrichment approach, where we thought that we could enrich based on mutation and slope. In hindsight, even an A5E carrier could have a life expectancy of 9 months or over 4 years. And if one is randomized to one arm and the other is randomized to another, then that's not balanced. And we know that the decline on the ALS-FRSR can be nonlinear. And so that, in hindsight, of course, is not particularly prognostic, at least on an individual participant basis. But I mentioned that there had been so much work prior to, and certainly in parallel with the conduct of Valor, to understand the behavior of neurofilament. And I mentioned that it has been shown to be prognostic in the broad ALS population for disease progression and survival. And the same was true in the context of the SOD1 population. So you can see here on the left-hand side, when we looked at pre-randomization ALS-FRSR slope on the top figure relative to the change in ALS-FRSR during the study period. And these data are from the placebo participants, so no confounding effect of any treatment. And then we look at the relationship between baseline neurofilament levels and the change in function over time. Neurofilament was a stronger prognostic tool than was pre-randomization slope. Again, standing here today, that makes a lot of sense, but at the time, perhaps not so much. And we can use neurofilament in several different ways. I think as of today, there's a lot more to understand about neurofilament and different factors that may impact levels of neurofilament that are unrelated to ALS disease activity. But we can immediately, based on the literature, start incorporating it to stratify populations, subgroup populations, or my personal favorite, incorporating neurofilament levels at baseline as a covariate to control for heterogeneity on an individual basis. The other was duration. I've alluded to this a couple of times, but 6 months was not long enough in the context of the tofersen program. We did design the study and calculate a sample size based on a limited amount of natural history data. Again, just sort of emphasizing the importance of really understanding the natural history, which is easier said than done. We both underestimated the time that it would take to reach maximum biological activity and the time that it would take for that biological activity to translate to clinical benefit. You saw that, again, neurofilament levels didn't reach their nadir until week 16, so expecting to see clinical differences in a 28-week study, in hindsight, would be quite a challenge. And again, we overestimated the declines that we would observe in the placebo arm. So we assumed we would see 24.7-point decline in that primary analysis population in the placebo participants based on what we knew at the time, and we saw an 8.1-point decline. So again, just emphasizing the importance of really understanding and making sound assumptions around what to expect in your control group. But we were able to do something about that because we had the open label extension ongoing. So the extension allowed us to look over the longer term. It also allowed us to zoom out, and instead of looking at these poorly enriched subgroups, really looking at everyone, at all 108 participants, and again, using neurofilament as a covariate to control for heterogeneity at baseline. And we were able to see that in the early start group, and I should emphasize that these are exploratory analyses, but in the early start group, across the board with function, breathing, and strength, we saw that those individuals that started tofersen earlier did better than those that started tofersen later at the time of the start of the extension study. So we have all of, safety profile, very important. So we also understood a bit about the safety profile at the time. We had a reasonable amount of exposure given the rarity of the population. And perhaps most important to note, there are serious neurologic events associated with tofersen. So in about 7% of participants that receive the therapy, that's both in clinical trials as well as our experience in the expanded access program, we're seeing events of myelitis and radiculitis, papilledema, increased pressure, aseptic meningitis. And to date, these events have been managed via standard of care. Most individuals have been able to stay on therapy, and those that have discontinued have experienced resolution, but it's something we're watching very closely and trying to understand both in clinical trials and in the real-world setting. So all of those data in hand, we filed an NDA, we submitted an NDA, I should say, to the FDA, and we submitted that NDA for accelerated approval. And so I thought I would take a minute to talk about why did we believe that that was the appropriate pathway? And what does that mean to pursue accelerated approval? And so I'm going to jump to this is reg speak. I'm going to jump to what's easier for me to interpret personally, which is to really understand if a surrogate endpoint is reasonably likely to predict clinical benefit, which is the bar that you have to achieve to secure an accelerated approval. It really comes down to two factors. The first is biological plausibility, and the second is empirical evidence to support that relationship. So we've talked about the biological plausibility in different parts of the talk today. We've talked about the fact that we understand the pathophysiology of SOD1 ALS and that this toxic protein is resulting in degeneration and death of motor neurons. And when that's happening, neurofilaments are released upon damage to the axons, and we're able to measure them in the blood and CSF. And we know that higher levels of neurofilament are associated with a more rapid disease progression and survival, as has been demonstrated pretty robustly in the literature. So there's biological plausibility that lowering neurofilament should be clinically important. But we also have to show that that's actually the case. And so I'll take a moment to talk specifically about what the tofersen program had to add to that equation to secure accelerated approval. We've seen the reductions in neurofilament in the Valor study. This is just showing that those reductions were consistent. This is always a question that comes up. Do you see the same thing with neurofilament light, phosphorylated neurofilament heavy? Do you see the same thing in CSF and plasma? In the case of the tofersen data set, although there is some variability and there are differences across assays, we do see very similar behavior. And in fact, with longer-term data that Dr. Miller shared at Niels a couple weeks ago, you see even sort of closer correlation between plasma and CSF over time. We saw that individuals that received tofersen in nearly all cases experienced a reduction in neurofilament, and so this was an important component of the study as are the conclusions as well. And this is a figure from the FDA, actually, that they shared at the PCNS Advisory Committee meeting that illustrates that these changes in neurofilament, these reductions in neurofilament, which you can see in orange, actually preceded any detection of a change in ALS-FRSR or difference on ALS-FRSR shown in black. But to really interrogate that relationship, we had to develop a surrogacy model, a model with a causal inference component that essentially accounts for natural disease progression. So when you're trying to interrogate the relationship between reductions in neurofilament and slowing of decline in clinical function or strength, you have to take into account what would have happened per the natural disease progression in that individual. And that's not easy to do with simple correlation analyses. We get this question a lot as well. So this surrogacy model essentially takes that into account. In my sort of simple brain, what it does is it creates a matched control for each of the tofersen-treated participants, and it estimates, based on data from Valor and the extension, what would have occurred had that tofersen-treated individual not received tofersen. And what the model tells us, it's quite complicated. The equations send my head spinning, but what the model tells us is that there is a relationship, that the reductions in neurofilament driven by tofersen at week 16, so that was the time point that those reductions reached their nadir, correlated with a slowing of decline in clinical function. We'll look at ALS FRSR as an example at week 28. And in fact, if you take an example participant, because it will depend, the higher their neurofilament level at baseline, the more opportunity there is for them to differentiate from their natural disease progression, so the greater difference you would observe. But if we take someone at the sample mean, so 97 picograms per mil, and we look at what a 55% reduction in neurofilament would equate to, and because that's what we observed in the Valor study, it would equate to a 4.11 point difference on the ALS FRSR. So this is the surrogacy model that, as of now, only includes data from the tofersen program, but my hope is that with time, we'll be able to build this out and really interrogate this relationship across the broader ALS population as well as additional therapies that lower neurofilament come through the pipeline. This relationship also exists in the context of survival. So again, week 16 reductions in neurofilament correlate with a reduced risk of death equivalent events over time. I will caution that we do have a limited number of death equivalent events observed in the study, so these data are relatively immature, but you see what you would expect to see, that just like the literature tell us that early reductions in neurofilament are correlated with, again, a reduced risk of death equivalent events over time. So what happened next? We went to an advisory committee meeting, and I'm just going to click through the animation. And the advisory committee was asked to weigh in on several questions. The first question was essentially, do the data support, do the literature support, does the underlying sort of biology support that neurofilament reductions are reasonably likely to predict clinical benefit in SOD1 ALS? And the committee voted unanimously in favor of that. They were also asked to weigh in on whether or not the data, in my words, were supportive of standard or traditional approval. We debate the language used here quite often. And this was a split vote trending negative. And then the final question they were asked to weigh in on is whether or not the benefit risk was positive in support of use of tofersen in SOD1 ALS. And the panel members they reached consensus that the benefit risk profile was considered positive. And so about a month later, we received approval of tofersen, accelerated approval of tofersen, and this approval will ultimately, hopefully, be confirmed via the ongoing ATLAS study. So this is the first, as far as I know, interventional presymptomatic study in ALS. And this is a study that's being conducted in SOD1 variant carriers, mutation carriers that have elevated neurofilament. So the treatment is being initiated when neurofilament levels go up, suggesting that there's underlying disease activity, underlying neurodegeneration. Happy to talk more about the design, but I think it's important to reflect on why is the study important. I think many of you will know that by the time we have discernible weakness, it's estimated that 50 to 80% of the motor neurons controlling that muscle or muscle unit are gone. And so likely intervening at the time of symptom onset may be too late, particularly in individuals with a faster progressing phenotype. And so ATLAS is really designed to understand if initiating earlier, and as of now, the earliest we can go back is based on those elevations in neurofilament. Hopefully one day we'll understand even more about the presymptomatic period. Does that actually delay the onset of clinical signs and symptoms, and or does it slow the decline in disease progression, I should say, over time? ATLAS is another global community-wide effort. We started the study in August of 2021. We've enrolled 102 of 150 participants to date, and this is 150 is the target for the natural history run-in. So these are participants that are willing to have their blood drawn on a monthly basis, to have their neurofilament levels monitored, and again, waiting to understand if their neurofilament levels increase, at which point they would be eligible for participation in the interventional study period. We're hoping to have data from this study as early as 2028, but that will depend on how quickly we enroll. I haven't been keeping track of time, but I'm going to just quickly close with learnings. Again, my favorite topic. We've talked about a couple of these, so I won't belabor the point, but I would say that just overall, tofersen has shown us that prioritization of these targets and therapies with strong scientific plausibility is really important, and doing as much early de-risking as we can is also important. If we didn't have that strong scientific plausibility with tofersen, I don't know what would have happened when the primary read out. I don't know how deeply we would have all as a community dug into those data and really tried to understand versus just assuming that the drug had failed. Use of improved mechanisms to control for disease heterogeneity. I talked a lot about neurofilament, and that's, again, something that can be used across the broad ALS population in this context. I would say that there's also a lot of other really important ongoing work. The NCALS prediction model is being optimized and things of that nature. So there's a lot more to understand about what factors or what combination of factors are most prognostic for disease progression and survival. I'm bouncing around. But pursuance of new endpoints and new biomarkers and making those decisions of what to put in your trial based on input from people with lived experience. And I would also suggest based on input from care team members that are not always at the table. There's some really interesting data that have been shared by the team at Washington University. Their physical therapy team, physiatrists are looking at endpoints that we never thought to look at, like the Berg balance test and the tug test and the 10-meter walk test. And they're looking to see, are they actually able to detect changes on those measures more easily than traditional measures like the ALS-FRSR. So I think that there's a lot that can be learned from our sort of cross-functional care team members. Consideration for the right duration study. I often get asked, is 6 months too short? In the case of tofersen, it was. A lot of that depends on the specific drug that you're developing. And if it takes a while, you have to take into consideration how long it takes to reach biological activity, and that needs to inform the duration of your study, as does the heterogeneity of the population, taking into account you may not see what you expect to see. But this is my quick soapbox moment plug for the importance of open label extensions. I think they should be present in all of the studies that we conduct in ALS. And finally, acceleration of time to intervention. The tofersen program in its totality teaches us even accelerating treatment by 6 months made such an important difference for those participants, and I would argue in many cases is too late to optimize potential effects of tofersen. And so the further back we can go, which again depends on our understanding of what is telling us that there is active disease activity in pre-symptomatic gene mutation carriers, for example, the better. And so with that, I will close by just thanking the study participants and their caregivers, their families, the steering committee members, investigators, site staff, this entire community, because again, not just enrolling these studies, but also sticking with us and really helping us understand what the data were telling us and where we went wrong. We have a lot more to do. We're just getting started, but I'm quite humbled to be part of this program and part of this journey and part of this community. So thank you.
Very nice. Can you talk about the FDA and how they view treating earlier and earlier and earlier? Because obviously that's where this should go.
Yeah, I won't speak on their behalf, but what I would say is that they're often pushing sponsor companies to consider this. I would say that what I have learned from conversations with the FDA, and I think that the legacy team has had direct conversations with the agency about this and has incredibly important input, is that they see this as one disease spectrum. They don't see it as distinct populations, symptomatic and pre-symptomatic. I think they very much understand the importance of intervening as early as possible, and they are continuously at the table, willing to have conversations. I would argue that the ATLAS study is one of the most unconventional studies full of scientific uncertainty that I will ever conduct, I hope. And it's one that I think that they have understood that scientific uncertainty, but been, again, there to discuss and to iterate and to react along the way.
Hello. Name is Wenshen Yang from Northeastern University. We work on SOD1 ALS as well, trying to develop small molecule to stabilize it. The question is, SOD1 as one of the major enzymes for superoxide dismutase has important role of protecting neuron cells from those superoxide species. Other than stabilizing only neurofilament, Is there any other effect that tofersen actually does to stabilize it and rescue its enzyme activities so that we have better functions?
Yeah, thank you for the question. I am a Northeastern grad, so it's nice to meet you. I would say the short answer is no, but it's in the works. So we're working with Peter Anderson and others to really try to quantify enzymatic activity and really try to answer this question. I think neurofilament from a sort of a true biological activity is the best tool that we have at the moment. The SOD1 protein assays that we're using, at least in the context of tofersen, they're measuring both sort of mutant and wild type proteins. So they give us some idea of the fact that we're reaching the target, but they're not telling us anything about disease activity, certainly not telling us anything about enzymatic activity. But it is something that we're keenly interested in understanding better and working on, albeit in a very exploratory manner at the moment.
Hi. Do you believe that you're achieving the maximum sufficient reduction of SOD1 to achieve maximum clinical effect, obviously dependent upon the time of intervention, because clearly earlier you probably could reduce less and still have an effect. Do you believe that you are achieving as good as you can probably get on this target with these ASOs?
Yeah, it's an excellent question that we think about a lot as well. The short answer is no. I'm not completely confident that it is. What I will say is that, again, it seems like a relatively modest reduction in the CSF, so about 35%. When we look with all of the grains of salt necessary, when we look at what that equates to through modeling based on NHP data, we estimate that equates to over 90% knockdown in the cord and in tissues of interest. And so we are achieving quite a bit of knockdown, again, with the caveat that that's based on extrapolation from preclinical data. We see something similar in the available autopsy tissue that we've assessed. So we are seeing exactly sort of what we would expect based on the NHP data we're seeing in these autopsy tissue, but it's quite limited at this time. So we're continuing to keep an eye on this. But I think, the truth is we don't know. But I think that based on what we're observing as of now, what we hope to do is intervene as early as possible such that we can optimize benefit. We wouldn't be planning to dose higher based on a number of different factors specific to tofersen.
And we'll just do one more.
Hi, Steph, over here in the back.
Sorry. Hello.
I'm Rick.
Thank you.
I'm not a familial ALS. I'm a sporadic ALS person, and I'm Canadian. And so I want to make a comment and then just ask a kind of a high-level question, because I'm not a bioscientist, although I'm slowly becoming one, I think. My friend Daryl lives in Vancouver, worked 16 months through Dr. Genge and other channels to get access to tofersen finally earlier this year, and he's in, I think, his 6th or 8th month of receiving it. And so on behalf of him, he's got SOD1. Thank you. Thank you to you. Thank you to Biogen and the whole team. I think it's amazing. And now all about me. In the findings or the learnings, there's a lot of stuff packed in there.
Yeah.
Where do you think the strongest place to pivot is for your team? And is it in the genetic or the familial side of the things, or is it to extend those learnings into something that can help the rest of us?
Thank you for the comment and the question. I would say, so real quickly, when Biogen was working with Canop over a decade ago on the dexpramipexole program, and the Empower study turned out the way that it was, and we could talk again for another hour about why that might have been, we decided to double down on genetically targeted therapies, because we thought that we would be able to learn about how to run the right experiments by learning from sort of, again, these programs with stronger scientific plausibility. I don't think anyone really understood how accurate that was, because we didn't think we would find ourselves in such a challenging position. But those learnings are being pulled into our pipeline every minute of every day. And so we have the ongoing program that is an ataxin-2 targeted ASO that's in a phase 1/2 study now, and in preparation for hopefully having data from that study next year, we're actively designing the statistical analysis plans with all of these learnings baked in. So how do we control for heterogeneity? We're going to incorporate neurofilament as a covariate, for example. And so we're constantly reassessing how we're running the trials for the broader ALS population in the case of BIB105 as well. And I see that happening everywhere. I see that all companies are learning and iterating and sharing those learnings. And it's a really special thing that's perhaps unique to the ALS community. So.